I'll just share my screen so that we can begin with this. So as I just stated, uh, we are going to like go through this session in two parts. The part one of the session will be dedicated to previous year questions pertaining to the week that we are discussing about. So this session is mainly about week two. So week one, I did conduct a session. I have actually posted a recording, but since NPTEL had forgotten to post the link for that session, I think you guys were not aware about it, but there's a recording that is available, and I'll be picking up after what I have actually talked about in that session. So at the end of the session, maybe we can also discuss if like significant number of you are willing. If you want, I can also conduct week one session if you are not just satisfied with the recording, right? So for now, we'll talk about week two, and pertaining to week two, there were some questions that were given for previous like 2023 as an assessment. Like you have been given an assessment task, and we are just going to go through that assessment. Before that, let me finally introduce myself. I am Arveen. I am a PhD scholar at the Department of Biological Sciences and Bioengineering at IIT Kanpur. And in both of these sessions, what we'll be doing is we'll revise whatever Professor Gupta has already gone through, and we will also discuss some questions or some extra pointers regarding that. So, without much ado, let us start with these questions. Yeah, just a moment. Yeah. So, question number one last year was, if a small positive current is passed into a neuron, the voltage of the neuron that is relative to outside will increase. Is this true or is this false? So I'll give you a moment to think about it. Thereafter, I'll come to the answer. Right. So since it's a true false questions, we can move to the answer. Uh, yes. So the answer for this particular question is true, right? So if positive current is passed, so this is how it was depicted, right? So there could be electrical stimulation that can be given to a particular neuron or set of neurons. For example, if we are talking about one particular neuron and we are injecting a positive current, right? So what will happen? There will be depolarization of the membrane. Why? Because we are saying that what is a polarized membrane? A membrane that is at its resting membrane potential. For instance, if we talk about squid, that resting membrane potential will be minus 60 millivolts, right? So over here, if a positive current is given, it will decrease, right? It will come towards positive side. So that phenomena is known as depolarization, right? So yeah, what will happen is that the voltage of the neuron will increase, right? Because it is moving towards the positive direction, right? So that is what is depicted in this figure that I have taken from Sir's lecture. And this basically says that if a small positive current is given, then it will increase a little, the voltage will increase a little, right? If it is continued to be given, it will keep continuing to increase a little, but only if a significant amount of positive current is actually given such that it actually crosses a threshold, which we'll talk about in the later weeks, right? So if it can cross a particular threshold, it will actually go to plus maybe 40 plus 50 volts, right? So that is basically generation of an action potential. So yeah, that is it for this question. Let us move to question number two. Question number 2 says, consider a cylindrical dendrite, right? So there is a cylindrical dendrite that you have and you cut it, just a moment. You cut it along the length at 2 places to get a cylindrical piece whose total membrane resistances are. Now you cut this again along the length in the middle to get 2 cylindrical halves, right? What will be the membrane resistance of each half? So they have given a resistance value of R when we have two cylindrical pieces of a dendrite, right? Then they are saying along the length. If these are two cylinders, then along the length we are cutting it again, and then they are asking what will be the resultant membrane resistance of one half that we will get, right? So what will it be? Will it be R or R by 2 or 2R or R by 4? So I would like you guys to think about it a little. Maybe if somebody can answer it, that is most welcome. If not, we'll obviously discuss the answer in a few moments. Okay, so nobody's willing to even be wrong, right? It's okay. So we'll discuss
I
the
think it
answer.
should be. I think it should
Yeah,
be R only.
it should be R.
Yeah,
Okay.
because it doesn't. Even if you cut it along in the middle of the two cylinder halves, the resistance should remain the same. That's what I think.
Okay. Anybody who wants to agree or disagree with that? Okay, so we'll see the answer, right? So the resistance is actually going to increase. It is going to double, right? Why is it going to double is, what is the resistance depending upon over here? So over here, resistance is actually depending on the surface area that it is provided with, right? So when the surface area will become half, when we cut basically the cylinder along the length, the surface area is going to reduce into half. And therefore, because it has less surface area now, so the current flowing will basically, you can think about it as if I had two basically cylinders that were fused into one, it would have a larger diameter, right? If it would have a larger diameter, current would flow through it with lesser resistance, right? Because the diameter is larger. Now, if I'm cutting it along the length, I have a narrower tube. Now, in a narrower tube, current is going to face a lot of resistance, right? So, because we are cutting it into two equal halves, it is going to double over here, right? So, yeah. So that is the answer, 2R. Coming to question number 3, a sodium-potassium exchange pump brings sodium inside the neurons, bring potassium ions inside the neurons, brings both sodium and potassium ions inside the neurons, or releases energy in the form of ATP. So which one do you think is the correct answer? And this is one of the theoretical questions pertaining to this course. So it has got to be one of the easier ones, right? Okay. So the answer for this one is that sodium potassium exchange pump basically brings potassium inside the membrane and pushes sodium outside the membrane, right? So that is how it is working. And it is definitely not releasing energy, it is utilizing energy, right? It is an active process. That means that it requires ATP to perform its function, right? It is actually a very energy extensive or energy expensive task that the neuron is doing to maintain its membrane potential, right? So this is a representation of the sodium potassium pump. You can see that the sodium potassium pump will bind 3 sodium ions and a molecule of ATP. This is inside the membrane is the yellow color and outside the membrane is the blue color, right? So from inside it is going to take up 3 sodium ions and it is going to take an ATP, right? And it is going to transfer it outside. Now, once it has reached outside, Na will leave this particular protein, and two key plus ions can actually come and bind to it, right? So the splitting of ATP will provide energy to change the shape of the channel. What is the change of shape going? Earlier it is like this, and because ATP is there and it is releasing energy, it will basically become like this, right? So that is the change in shape that is being talked about. Then the sodium ions are driven through the channel, right? And sodium ions released to the outside of the membrane. Then the new shape of the channel, that is this, is basically favoring intake of potassium ions. So now potassium can come and lodge into this particular channel, and this phosphate, this potassium can actually be released into the membrane, right? So that is how it is working. And it is utilizing ATP as the energy currency. Coming to question number 4, it says, consider a neuron at rest. Okay, let's say the equilibrium potential of the sodium ions for that neuron is E. All right. We somehow magically swap the inside and the outside concentration such that whatever concentration was present inside is now outside and vice versa. Right? What will be the new equilibrium potential of the sodium ion? So will it remain as E, or will it be 1 over E, or it will be minus E, or it will be E by 2? So what do you think will happen? Go through the statement again. Maybe it will come to you guys. Okay. So what is happening over here? What they are saying is basically the neuron is at rest, and then they have thought of a hypothetical situation, and they have said that when it is at rest, the sodium concentration or the equilibrium potential of sodium is E, right? And then they are bringing a hypothetical situation wherein they have said that they have swapped the concentrations inside the cell and outside the cell. In that case, now what will be the concentration of sodium, right? So if it was E initially, while it was at a resting membrane potential, and the conditions have now been swapped, it will account to be minus E, right? Why? Because it is just the reciprocal of what it was happening. That is, if the concentrations are switched in the Nernst equation. Yeah. Does anybody want to say anything? Okay, I think that was not on intention. Yeah. If the concentrations are switched in the Nernst equation, right, which is basically determining how much difference is there for a particular ion inside and outside, right? And what is the driving force for a particular ion? The ratio of the concentration inside the log becomes inverted or reciprocal, right? Therefore, the final values will be negative of the original value. That is, if the original value was log A upon B, the final value will be minus log B upon A, right? So that is why this answer is going to be minus B. Yeah. Coming to question number 5. In a typical neuron at resting state, which of the following is most accurate, right? Potassium ions would like to move from inside to outside according to the electrical gradient, or potassium ions would like to move from inside to outside according to the concentration gradient, or potassium ions would like to move from inside to outside according to both the concentration and electrical gradients, or the potassium ions would like to move from inside to outside by none of the gradients. So I think that much time was enough. Me reading and you thinking about this question, right? Because again, it is a simple one. So the answer for this one is that the potassium ions would actually like to move from the inside to the outside according to the concentration gradient, right? So we know that the sodium ion concentration inside and outside the cell, like with respect to each other, is more outside, while in case of potassium ions, it is the inverse of that. The more concentration of potassium ions is inside or within the cell, right? So that is why, since potassium ions are present in higher concentration inside the cell, therefore they would like to go outside according to the concentration gradient. According to the electrical gradient, however, they would like to move inside as the inside potential is negative, right? So according to the electrical gradient, if we think, then both the sodium and the potassium ions, all the positively charged ions, will follow like this path from outside to inside, right? Because inside the resting membrane potential is has a negative value. So this is going to be true with respect to concentration gradient, right? Then we come to question number 6, which is myelination of an axon increases its length constant. Is it true or is it false? So this I would really like somebody out of you to answer. This concept has been taught by Sir. So just saying, I have been in Sir's classes, and at least his way of teaching is to get people answering or interacting in the class, right? So that all of you are attentive. Because he will not move ahead until and unless people are answering, right? So I have attended these lectures offline, and that is how Sir likes to do it, at least. Okay. Yeah, I just admit these people. Hmm. So no answers for myelination of an axon increases its length constant. Is it true or is it false? Okay, maybe I should have done the revision class before the previous year questions. So in that case, I would have actually revised this with you guys and you would have been answering. That is at least what I'm assuming. Okay, so this actually is true. Why is it true is because what is the length constant? So if you remember from Sir's lectures, length constant is basically lambda. And what does lambda depend on? It depends on two kinds of resistance, Rm and Ra. That is the membrane resistance and the axial resistance. Right over here, if there is myelination of the axon, that means that the membrane is being insulated, right? So the membrane resistance is going to uh, increase, right? And since Rm, since lambda is if there is more membrane resistance, right, then there will be less leakage of whatever is flowing through the this particular cylinder or axon, right? And thus lambda will be bigger. So for example, it is written over here, more Rm will pertain to less leakage, and that will result in bigger lambda or bigger length constant. So yes, myelination of an axon will increase its length constant. And this is actually how humans have developed their neuronal system, right? So because unlike the squid, we do not have axons that are very large, such that they are going in orders of millimeters. We have very very tiny axons in the order of microns, but yet we also do have to conduct signals at long distances, which is why basically what humans have done is that they have been able to, or their system has been able to come up with this phenomena known as myelination, which basically insulates the axons of their neurons so that the conduction is better, right? Yeah. So which of the following is the most reasonable value of time constant in neurons? So is it about 10 minutes or 10 seconds or 10 milliseconds or 10 nanoseconds? Okay. So the answer for this one is going to be 10 milliseconds. Why is it so is, what is the time constant? Now the time constant is tau. Now this was a value that you might recall from the time constant lecture that was there in this particular module, right? And what is the value of tau? It is basically resistance over capacitance, right? And this is something that sir had discussed in that lecture also, that if we, yeah, okay, there's some noise that's coming. I will just mute. Okay. Yeah. So in that particular lecture, also we had discussed that if we talk about neurons, for instance, then in neurons, the value of resistance is going to be in the order of mega ohms.
I can't hear you. Ma'am, you have muted. I can't hear you.
Oh, okay, I am sorry. I hope that just happened. Uh, okay, I am just going to repeat this particular question so that yeah. So what I was saying was that uh, time constant is basically tau, and tau is equals to resistance into capacitance, right? And resistance in a neuron will be in the order of mega ohms, that is 10 raised to power 6 ohms, while capacitance will be in the order of nanofarads, that is 10 raised to power minus 9 farads, right? So the resultant tau value will be in the order of 10 raised to power minus 3 seconds, or it will account to almost 10 milliseconds. Right. So that is why this is the particular answer for this question. Okay. I hope I was audible in the previous question. So, okay, I'll move on to question number 8. Uh, yeah. So consider a long cylindrical axon whose membrane potential is minus 60 millivolt at rest, right? And the length constant for this particular axon is 2 millimeter. A constant hyperpolarizing current is injected at point A in the axon. When the current is maintained for a while and a steady state is reached, which of the following points in the axon will have the least negative membrane potential? Okay, so I hope you guys can actually go through the question part again. Let me just go through the options once. It is exactly at point A, or 2 millimeter left of A, or 4 millimeter left of A, or 5 millimeter right of A. So just read the question once again. Then I'll discuss the answer. Yeah. So I hope you all have gone through the question. Now let me break the question down for you. Right. So what are the pointers that they have given to us in this question? One is that they have said, yeah. One is that they have said that the axon is at resting membrane potential, and its resting membrane potential is minus 60 millivolt. The second thing that they have told us is the length constant, that is 2 millimeter. Right up till here, it looks like somebody's uh, numerical is going to come up in this question. But in the next line, what are they saying? That uh, a constant hyperpolarizing current is injected at a point, right? Now, what does hyperpolarization means? What is the normal polarization value? So if this is the graph and this is positive and this is negative in case of this polarization, so at minus 60, right? I'm sorry, that is what my mouse is doing. Uh, what I tried to write is 60, right? At minus 60 or at this particular point is where is what is the polarized value or the resting membrane potential, right? Now, depolarization would mean moving in this direction or the positive direction, right? And hyperpolarization will be moving in the opposite direction, that is in more negative direction, right? So what are they saying now is that they are giving a constant hyperpolarizing current. So it means that they are giving a more negative current, right? At point A, where basically that electrode has been injected, right? So if we say that this is an axon and this is where the electrode has been injected, so this basically is going to be point A, right? And they are going to, they are what they are saying is when current is maintained for a while, so it is constantly being given the hyperpolarizing current, so it will go more and more negative, right? So more and more in this direction it will go, and a steady state is reached such that after a point it will go no further, right? Maybe it reaches till around minus 80 or minus 90, and below that it can't go more negative also. So at this particular point, which point in the axon will have least negative membrane potential, right? So exactly at this point A, or maybe 2 millimeter left, 2 millimeter left of this point over here, right? Let us say this point as B, or 4 millimeter right of it, let us say at point C, or 5 millimeter right of it, let us say at point D, right? So which one of these will have the least negative membrane potential? That is the question. So it did not turn out to be a numerical actually. It is more of a logical question, right? So if you recall, the length constant is going to basically veer off as we go away from the point of injection, right? So the farthest point over here, that is 5 millimeter, will actually be the correct answer, right? So let me show it with the depiction that I have not kept that figure. So let me just move back to this. So if we are saying that a current is coming over here, right? So maximum current will be at this point. As it goes over here, it will dissipate a little. As it goes further along, it will dissipate furthermore, right? So the current reaching the farthest point will be the least, right? So it will be the least affected. So that will be the least negative, right? Because over here, negative current is being given.
Ma'am,
So
why
I
it
hope that
is
was,
not? Ma'am, why it is
yeah,
not 4 mm right of it? Why it is 5 mm? How does that
because
differ?
5 is the farthest point, right?
Okay. Wait, okay. It will go
Yeah.
farthest. Got it.
Yeah, so it was the farther that we are going, like right and left over here make no sense at all, right? Even if the options were 5 right and 5 left, both of them would have been correct, right? So
Okay.
that is not the point. The point is whatever, like for example, if I am speaking in an auditorium, the person who is closest to me can hear me the loudest, but the person who is sitting far away or at the end of that auditorium, like provided that I'm just speaking with the aid of no mic, right? No microphone, no speakers are helping, my voice reach that point, right? So the person who is sitting at the back will be able to hear me the least, right? So that is the same phenomena or the same logic that is applying over here.
Thank you, ma'am.
Yeah. So let's see what is written also. As the injected current, yeah. As the injected current is hyperpolarizing or it is negative, the membrane potential at point of injection A will be most negative. The effect will reduce as we go farther away from A. So the farthest point will have the least negative membrane potential. Now, also, another disclaimer that I wanted to give to you guys is why I have written down each of these statements is, sorry, because these presentations also I will share with you, right? So even if you are going through them, so that like without the discussion part also, I want you to understand why we are saying this is the answer of this particular question, right? So that is why it has been written also, just in case you are going through it again. Okay. So now question number 9. Consider a neuron is in the default resting state. Now, a constant current stimulation is applied in that neuron, right? In the initial period of stimulation, which of these will be true? Current will mostly charge the membrane. Current will mostly flow through the ion channels. Current will mostly charge the ion channels, or current will mostly flow through the ion exchange pumps. So, which of these is going to be true? Let us look at the answer. Yeah. So current will mostly charge the membrane, right? So once, why are we saying this? Because they have specified that in the initial period of stimulation. So if you would remember when Sir was discussing this particular, circuit, and in the point in over here represents inside the membrane and out over here represents outside the membrane, right? Or this particular value represents the resistance and this particular is the capacitance, right? So initially, if we stimulate it from inside, right? From inside over here, there is some kind of a stimulation. Since both of these potential or voltages will be zero, all the current will flow through capacitance, right? So none will flow through resistance and all of it will flow through capacitance. And in case of capacitance, it has the capability of accumulate charge at the capacitor, right? So it will not let the current flow through, but it will accumulate the charge. So that is why we are saying that in the initial period, when there is no current that is going through the resistor, but all of it is going through the capacitor, it will mostly charge the membrane, right? So the capacitive part of the membrane offers no resistance and current will initially go through the capacitor to charge the membrane. I hope that is clear. Coming to question number 10, which is the final question for this first part of presentation, which is the resting membrane potential does not depend on which of these following things. Is it the temperature or the concentration of ions with zero permeability, with non-zero permeability, or concentration of ions with zero permeability, or whether the ion has positive or negative charge? The keyword over here is not. It is not depending on which one of these following things. Okay. So the answer for this one is that it is not going to depend on the concentration of ions with non-zero permeability, right? So if the ion is not going to pass through at all, how will it depend on the resting membrane potential? While on the other hand, the ions that are going to pass through or have a non-zero permeability will affect the resting membrane potential. Similar is the case with ions that have a positive or negative charge, that is also going to affect it. And temperature can actually change or alter the composition of the membrane, or how densely or how loosely it is packed up to a certain extent, right? Also, there are some mechanically gated or sensing ion channels that are present, which can also, open or close with their response to temperature. So that is why temperature is also going to affect the resting membrane potential. Yeah. So that is it for the first part. The ions that are not permeable do not affect the membrane potential. This can also be seen in the GHK equation, where the ion concentrations are multiplied by permeability, and therefore ions with zero permeability will not have any effect, right? Yeah. So what we'll do is we'll begin with the second part. Yeah. Just allow me a brief water break. and we'll begin with this after that, right? So what we'll be doing in this is that we'll be revising the concepts that were taught to you this week. Also, in the first week's discussion session, like I said, the one that I have recorded and I have handed over to NPTEL, uh, in that particular session, I was not able to complete the entire week one's syllabus. So, I will be starting with brain orientations, that module over here, and I'll be going up till the length constant module, right? So, the equilibrium potential that we'll be discussing in the following week, like next Sunday, you'll have a class again. So, that we'll be discussing at that time. So, from the brain orientation to the length constant, that is what I'm going to recap for you guys. So, please allow me a short break. Yeah, so just to ensure that everything was fine, I'll just share my screen again, right? Um, because often in these things, it causes trouble once I switch to another presentation. Yeah, so I'll be sharing my screen again. Um, so I hope it is still visible to everybody. So as I just briefly told, what we'll do is a recap of a part of week one module and a major part of week two module, right? Also, if you have any doubts, I'll try to address them. So you'll have to speak up for that, right? As we are going through those concepts that you have doubts in, you can speak up. I will try to address them. If I am unable to do that, uh, so I will surely read up upon it and I will discuss with other people in the lab and with Sir maybe, and I can get back to you with the most probable answers for those. Right? So yeah, so let's just begin. So when I said we'll be going to start with the brain orientations module, like we'll not go through the entire lecture. As such, what we'll go through is what was the gist of that lecture, right? So, for example, in this particular slide, most of the things we can understand that have already been taught to you. So, we'll just brush it up, right? So, when we talk about brain orientations, why are we discussing this subject at all? So that we can basically refer to different regions of the brain, and so that if when I am referring to it, you can understand it, right? So, for example, if I am talking about a particular region that has a name, for instance, medulla oblongata or cerebellum or cerebrum, for instance, right? Then you can anyway understand it. But if, for instance, I am talking about a region that is between maybe the cerebrum and cerebellum, right? Now, you might not in your head always remember like where these two regions are lying and what is exactly between them and what is away from that. What does this person mean when she says left of this pineal gland? What does she mean when she says right of pineal gland? Right? So, first of all, you'll have to know where pineal gland is for that, right? So, that is all not always possible. So sometimes we will go through some generic terms as to how we can basically uh, talk to somebody about certain regions of the brain, right? Uh, and your MRI, CT reports also often, name brain regions. based on these terminologies, right? So when we talk about these first two terms, that is anterior and posterior. What does anterior mean? Anterior always means ahead and posterior always means towards the back, right? So when we are talking about humans, the region of the brain that is towards my face is the anterior region, and the region of my brain that is towards my back, right? That is the posterior part, right? And then what is superior and inferior? Again, the one that is at the top is superior, and the one that is at the bottom is inferior, right? So how what will it be? The one over my head is the like close to the top of my head is the superior region of the brain, and the one that is closer to say my neck is the inferior region of the head or the brain, right? Then there are some other terminologies that we can discuss, say dorsal and ventral, right? So dorsal and ventral is similar to superior and inferior in case of the brain, right? And initially, when dorsal and ventral came, like these two terminologies came into existence, the then we were not actually bipedal animals, right? So, for instance, if you talk about a dog or a cat or any animal that is not bipedal, right? In them, dorsal side always represents the side that is facing the sun, and ventral side is always representing the side that is facing the earth, or it is facing away from the sun, right? So naturally, for us, the back side is dorsal, right? From the skull till the back, the entire thing is dorsal, right? So it starts at the top of the skull, while the inner side, right, is the ventral side. So the brain has some region like it is present just above my eyes over here also, right? That part is the ventral part. Okay. So that is dorsal and ventral. Then again, rostral and caudal is same as anterior and posterior. The side that is facing the that is to side of the brain that is towards my face, which is the anterior side, as we discussed, is also known as the rostral side. Right. The other that is the posterior end is also known as the caudal end. Right. That was that. Right. Apart from that, there are two more major terms that we should know. One is a medial, which refers to the middle, and lateral, which is anything away from the middle, right? Okay. So that is how orientations are in general discussed, not just in the case of brains, but in case of other body organs also, right? Then something that is very specific to brain over here is going to be how, over here I have also included what we have discussed about. So everything over here is written also. So once you get hold of these slides, maybe you can go through them also. Now, this is what we generally talk about when we speak in terms of the brain anatomy only, right? So, the brain, when you take a brain, you can face it in different, planes, and you can cut these brain into sections in different planes, and depending on which plane you are cutting making a cut in, these names will differ, right? So the first one that we will talk about over here is the frontal or the coronal plane. Now this is one of the most common planes in which basically when we study about, say, dopaminergic neurons that have some, that are depleted in conditions like Parkinson's, then we know that those are going to be present in the hippocampal regions, right? And sorry, in the dorsal subiculum regions, and so that we can look at both sides of the brain, both the dorsal subiculum region. What we are going to do is, generally people do a coronal cut, that is a cut along this plane, right? So once it is cut, you can stain that section and you can see like what is the neuron look like, how is the brain morphology, etc. So there are multiple things that can be done. Then the second most common one is the sagittal plane, right? Now, in the coronal cut, one cannot really look at hippocampus that well. And you might recall from Sir's lectures that hippocampus is one of the most studied regions of the brain, right? And then even the cerebellum, right? So over here in the coronal or the horizontal plane, the hippocampus, that morphology, the C morphology with the CA1, CA2, CA3 regions, if you are aware of it, like this morphology is not that visible. And this tree-like morphology of cerebellum is also not visible. So to visualize this morphology or visualize specifically the cerebellum or the hippocampus and even other brain regions, sagittal cuts are actually preferred. Right now, sagittal cut will be along this plane. So along the corpus callosum, if a cut is placed, then that will be the sagittal cut along a sagittal plane, and it will be known as a sagittal cut. Right? Then comes the horizontal cut. Now, what is a horizontal cut? When uh, there is a cut along this plane, right? Coronal was along this plane, sagittal was along this plane, and now horizontal will be along this plane. So it is kind of like a like in that rostral to caudal or anterior posterior axis, you are going to put a cut. Now that is going to be a horizontal cut, right? So these are the various morphologies in which the brain is actually visualized. um, so that is that. Let us now move into the details of the structures of the brain. Now, when we look at a more broader perspective, the structures of the major structures of the brain are these three, that is the forebrain, the midbrain, and the hindbrain, right? When we look into detail, we will see that there are multiple sub-regions that each of these major structures have, and we are going to discuss some of those sub-regions as well. So over here, at least you can make out that forebrain is the biggest part of the brain. Then midbrain is a very small region that is present between the forebrain and the hindbrain. And hindbrain basically includes those regions towards the posterior or the inferior side, and it includes cerebellum, brainstem, pons, medulla, etc. Right? So, so the structures in the forebrain, this portion that is yellow in color, will include cerebrum, thalamus, hypothalamus, pituitary gland, limbic system, and olfactory bulb. Okay. The ones in the midbrain over here, this pink region, will include various cranial nerve nuclei, right? So, cranial nerve nuclei means the cell bodies of the cranial nerves, right? Tectum, tegmentum, colliculi, and crura cerebra, right? And the hindbrain will basically include hindbrain, which is also, by the way, referred to as brainstem, will include the medulla, the pons, the cranial nerves, and the back part of the brain that is known as cerebellum, right? So let us look at each of these structures in further detail. Yeah. When we talk about the anatomy of the brain, the major parts of the brain are made up of different structures that each have important and different functions, right? So, for instance, all this hypothalamus, cingulate gyrus, corpus callosum, thalamus, all these are part of the forebrain, the pineal gland, and pituitary is also a part of this particular region. Then pineal gland is a part of the midbrain. Then midbrain separately is also depicted over here, right? And pons plus medulla along with brain stem is along with midbrain is known as brain stem. Then the cerebellum comes in that hindbrain region, right? So let us begin with forebrain. So the first, one of the most important parts of forebrain is thalamus, right? So thalamus is present near the midline of the brain, and it is known as the relay center of the brain. Why is it known as relay center of the brain? Because all the sensory information that is going into the brain, except for your olfactory sensory information, all of it is going to pass through this particular region that is known as thalamus, right? And along with that, whatever motor information is going out of the brain will also pass through this particular region that is known as thalamus, right? Then comes hypothalamus. That is located just below the thalamus and above the pituitary glands, right? So this is hypothalamus. This is thalamus. Between the thalamus and pituitary, it is located. And what it does is it basically controls the pituitary glands, right? So there are hormones that are secreted by hypothalamus that are going to regulate the hormones that are going to be secreted by the pituitary glands, right? And it will also be responsible for the body temperature as well as motivational behaviors like hunger and thirst. So the hunger center and the thirst center of the brain, these responses that our body is basically sharing is with the hypothalamus inside the brain, right? So another part in the forebrain is the hippocampus, which is part of the limbic system. It basically is located in each temporal lobe, right? So both the sides temporal lobes will have the hippocampus, and it is critical for learning and memory formation as well as for spatial navigation, right? Then comes amygdala, which is also a part of the limbic system and is located at the end of the hippocampus. Also, it is responsible for the response and memory of emotions and especially of the emotion that is fear. Then basal ganglia again is important for regulation of various motor functions. Yeah. And then comes cerebral cortex, which is the outermost and the top layer of the brain. It is also forming the major region of this forebrain, right? It is consuming the maximum portion of this forebrain, and it can be divided into these four regions that we will see later: the frontal, parietal, temporal, and occipital lobes. The cerebral cortex is responsible for thought processes like speech and decision making, and each different lobe is responsible for different kind of cognitive processing and functions, right? Also, both the cerebral cortex and basal ganglia are together known as cerebrum, right? Then there is one other region in the forebrain, yeah, which is known as corpus callosum, right? It is located above the thalamus and under the cortex, and it connects the right half and the left half of the brain. And it is responsible for communication between these two hemispheres of the brain, right? So, like if you would have heard of this very famous book known as Phantoms in the Brain. Um, I am really bad with names, which is why I'm forgetting the author of that book. He is a neuroscientist. He's an Indian origin neuroscientist who lives in US, who has written that book. And in that, uh, he has described patients who were suicidal just because of the disconnection of the two halves of the brain, right? So one half would have the suicidal tendencies, and the other half, although it was like just like a normal person, uh, these two halves, since they were not able to communicate, this person had a split kind of personality, and like it was uh, like unintentional for at least half of her brain that the other half of the brain was trying to kill her. I know it sounds very haphazard the way that I'm explaining it, but it has been written very beautifully in that book. So there are some cases wherein the connection between the left half and right half of the brain is gone. So in that case, even if I am thinking something with my right half, my left half of the brain will not be aware of that, right? So
Ma'am,
that
is
was
it a book
actually
by Sandra? Is it a book by Sandra and Ramachandran? I mean, both two writers, two authors?
yeah, I think it is.
It's not written by
Um,
one author, correct? Phantoms.
Phantoms in the Brain. Yeah. I think it is that. It is Ramachandran, but I do not remember the first name of like
Okay.
that
Thank you.
particular
Just
author.
trying to find out. This is worth reading a book. That's what you said.
Yeah. Uh, so that has this particular incident described in much more like reading from there will make much more sense, right? I might not have been able to justify that. I am very sure about that. So I'll just move forward with the lecture, something that I will be able to probably justify. So let's talk about the midbrain. So midbrain is involved in vision, which is particularly the visual reflexes, hearing, sleep-wake cycle, alertness, and temperature regulation, right? And pineal gland, which is also like it had been listed as the seat of intelligence by some of the primitive philosophers that we had studied about, right? That particular gland is also actually located in the midbrain. It is located deep in the center of the brain and in the midline between the two hemispheres, right? The part of the endocrine system produces melatonin, which helps maintain circadian rhythm and regulate reproductive hormones. Also, it regulates other endocrine functions and converts signals from the nervous system into endocrine signals. Okay. Then we come to the hindbrain. The first part that we are going to discuss about is the cerebellum, which is located in the lower area of the brain behind the pons, and it is responsible for balance and coordination of muscles of the body, right? Then comes the medulla oblongata, right? This part, which is located in the lower part of the brainstem and is responsible for maintaining vital functions of the body and involuntary actions that sustain life, like breathing, blood pressure, heart rate, swallowing, transferring messages from the brain to the spinal cord, etc. So all the involuntary functions that are happening in our body is basically controlled by medulla oblongata, right? Then comes the pons, which is located just above this medulla, right? And it is responsible for sending signals or messages between several areas inside the brain, especially between upper and lower parts of the brain, which plays a role in sleeping and dreaming, right? So, like we had talked about when briefly when we were talking about the cerebral cortex, there are these four major areas in the cortex: the frontal lobe, parietal lobe, temporal lobe, and the occipital lobe. Right, and we will briefly look at what their functions are. Also, if you look at them in a different angle from the top, what you see is the frontal lobe, the parietal lobe, and the occipital lobe. And since this temporal lobe is a little towards the bottom of the brain and the lateral, like how will you call it using those brain orientation? So it will be towards the posterior lateral side, right? So that is why it will not be very well visible from the top angle, like when you are looking at the brain from the top. Uh, Also, let me briefly introduce these folds and the depressions and the hills that are present in your brain, which are the sulci and the gyri, which are basically responsible for increasing the surface area of the brain. Right, so that there could be more number of neurons that can be packed up into your brain. So, like we said, we'll briefly discuss the role of these regions of the cerebral cortex. The frontal lobe is majorly associated with the speech and with motor functions, right? The parietal lobe is associated with a lot of sensory functions, such as, reading, that is the vision, and somatosensory association, and it is where the cognition or processing of somatosensory signals take place. It is also associated with the sensory information like speech and taste, right? Then the occipital lobe is a major visual area, like over here the vision and associating the processing of that vision is basically going to take place. And then the temporal lobe is majorly responsible for sense of smell and hearing and auditory association area, right? Why? Because it is kind of closer also to the ears and to the nose, right? Like that contact has to be minimal, right? So temporal lobe is associated with these functions. As was already, told to you by Sir, these are not the only regions that are going to perform these functions, right? And these regions are not only going to perform these functions also, right? So it goes both ways. So these are the major functions that they have been associated with, which does not mean that no other brain region will perform these functions. And it also does not mean that these particular brain regions are not going to perform any other function, right? Uh, Let us briefly discuss the somatosensory and the motor cortex. So the somatosensory cortex is basically over here, like you saw, right? These two strips. The motor cortex, which was majorly a part of the frontal lobe, and the somatosensory cortex, which was majorly a part of the parietal lobe. Right. So when we talk about somatosensory cortex, it means basically the region that is receiving the sensory stimuli and basically also processing that sensory stimuli in the brain. Right. So if we talk about this blue strip throughout the brain, which is a part of this parietal lobe, you will see that uh, the region within this blue strip that is allocated to different body organs is like not very proportional, right? So over here, like you see the abdomen and the digestive tract it has almost as much area as the tongue and the mouth together, right? And the entire face has much more area than the entire abdomen, although they would uh, almost be of the same size, right? And the leg, head, etc., that are of a much bigger proportion size wise than these areas. They have so less area that is dedicated to them. So that is how basically sensation works. So the parts that have more sensory stimuli to be basically felt up, they have more region dedicated in for them in this particular somatosensory cortex than the regions that have lesser basically sensory stimuli that has to be perceived, right? Similar goes for motor cortex. The functions that perform more motor functions, for example, your larynx, pharynx, you're constantly speaking, you are constantly breathing, right? You are constantly talking, you need your tongues, jaws, face, etc. to do that. You are constantly making different facial expressions. Your face is moving a certain way, your lips are moving a certain way, your hands, your fingers are doing a lot of function. Your hands, arms per se are not doing that much, right? Your fingers are working a lot. So these uh, uh, basically regions will have more motor area, more motor cortex area that is dedicated to them as compared to the, say, the trunk, the shoulder, legs, etc., that are not performing that big of a function, right? And these motor and somatosensory cortex, like which areas are mapped to them, was basically determined based on the works of this Canadian American doctor known as Wilder Penfield, who was working with a lot of epileptic patients. And like whenever there used to be a lobectomy, um, and he used to like have this cranial window in the skull of those people. He used to like, even if it was an accidental stimulation or purposely stimulating the brain, he used to always note down that, okay, when there was an electrode that went into this region, this region in the body got stimulated, etc. So his notes and his insights have helped people discover or allocate these particular regions to these particular somatosensory and motor cortex over here, right? And this is a artist's representation with regard to it. It's the sensory homunculus and the motor homunculus. Uh, uh, Depending on how much area in these two particular cortices is actually allocated to which particular region, right? Okay. Uh, so before moving to the next module, let us also go through the human brain waves. Right. So there are five major types of human brain waves that are present. from the decreasing order of magnitude of frequency. They are delta, theta, alpha, beta, and gamma, right? So gamma has the maximum frequency. It is when the brain is most active or alert, right? It has a person has heightened perception. They are learning or problem solving. They are performing problem solving tasks, or they are doing some cognitive processing, right? Then second to it is beta, which basically signifies the brainwaves of a person who is majorly awake or alert or conscious and is thinking, or the person is excited by some fact, right? Then is the alpha wave, which is when a person is mentally and physically relaxed. Then comes theta, which is brain waves just between 4 to 8 hertz. And this is when a person is basically either dreaming, or they are in deep meditation or reduced consciousness, or during deep states, creativity, or when they are having some insights, right? Then the final waveform pattern is a delta wave, which is just between zero point 0.5 to 4 hertz. And this is when the person is deep in sleep, right? Which is a non-REM sleep. A person is not dreaming. There is loss of bodily awareness, and repair functions are going on in the body and the brain, right? Uh, then we move on to the electrical activity in the brain, which begins the second module of the course, right? So we know that signals have to be transduced across neurons in the brain, right? Now, to transduce these signals, there is something that has to be done, right? How will it be transduced? So we know that it is transferred within a neuron and between two neurons by either electrical or chemical, or both these signals in combination, right? So over here, first we are going to look at the electrical portion of this signal transduction, right? So, this is an example of the neuron's electrical activity, which is a sequence of spikes, which is also known as action potentials. So, much later in this course, when we actually have discussed the equilibrium potential, will we discuss what exactly is action potential. So, since I have not kept that in today's discussion, we'll probably do it next week, right? Um, but like, forget about what exactly action potential is until we actually finally discuss it. This basically depicts some kind of a signal that is or activity that the brain is undergoing, right? So there are certain questions that arose, and sir discussed it with you. The first was, what gives rise to electrical activity in the brain, right? So it has to be exchange of or the movement of some kind of charge, right? So it that is the answer. It is caused by the movement of charged particles. The second was, what could be the charged particle that is involved, right? So these are basically ions, and these are ions that are commonly found in our body: potassium, sodium, chloride, calcium, magnesium, etc. Right? Then what makes the ions move in the brain? So there are certain gradients, both electrical and concentration gradients or chemical gradients, that are present for these ions. Like, they will be more abundant some place and less abundant some place. And these gradients will actually make them move, right? So these concentration gradients and electric fields make these ions move, right? These are the typical ionic concentrations in the mammalian cells, wherein the major monovalent ions are the sodium, potassium, and chloride, right? Sodium and potassium, you can see sodium is has a much more concentration outside the cell than inside the cell, or within the cytosol. Potassium, on the other hand, is more abundant inside the cell or the cytosol than outside the cell. Chloride, again, behaves much like sodium in its extracellular versus intracellular concentration, as its extracellular concentration is much more. Then there are two major divalent ions that are majorly found in humans, or our cells. That is the calcium and the magnesium ions, right? Both of them are present in abundance outside. the cell, right? So, yeah, so over here you could have seen that there is much more abundance of the positive charge outside the cell than within the cell, right? Due to which, again, the membrane, resting membrane potential will be negative. We'll come to that in a couple of slides. So over here, the first question is what prevents these ions from moving through the membrane and balancing out, right? Why are they just not going like across the membrane? Because the membrane is not freely permeable, right? That will not allow charged particles to go through, right? So lipid bilayer membrane is hydrophobic and it prevents ions from crossing, right? Yeah. So how do ions cross this membrane at all? It is possible due to presence of ion channels, right? So there are special protein channels that are present so that these ions can actually pass through. And they are specialized passages that are selective for particular ions and let them pass through the plasma membrane, right? So these are the types of ion channels that are present in our body. The first two actually depict what are the components that they can be made of. They can be made of different kinds of proteins, like a heteromeric protein that is coming together and forming a ion channel, right, through which ions are passing, or the same kind of components or homomers that are coming together and forming the channels. The first two are those, right? Then comes the gated channels, right? So the channels could either be voltage gated, that only at a certain voltage will these channels open up. Below or above that voltage, they will not be open, right? They will be closed like this. So physically, they'll be closed so that no ions will be able to pass through. Then are the ligand gated channels. Ligand gated channels basically require a ligand or a substrate that will come and attach to some part of this channel, which will be the receptor. And only when this receptor actually comes in touch with the correct ligand or the correct chemical molecule, will this channel become open? Otherwise, it will remain closed like this, right? The other could be the mechanically or stress gated ion channel, right? So this means that maybe this could be pressure gated channels or temperature gated channels, wherein only when a certain pressure is applied, then only these ion channels open up and they allow ions to pass through. And the fourth type is the always open or the leakage channels, right? So these have no closed state. They are always open and some amount of ions will always be going in and out of these channels, right? But these channels will also be specific for ions, right? There will be specific sodium leakage channels. There will be specific chloride leakage channels or specific potassium leakage channels, right? From the potassium leakage channel, sodium ions will not be able to go through. Similarly, from chloride ions, no other, ion, sodium, potassium will be able to go through, right? And leakage channels usually transfer the ions along their concentration gradients, right? Yeah, most of these ion channels are doing that only. The other is a sodium potassium pump or the ion pumps, right? That we will come and talk about later. So once the ions flow through these channels, how will the concentration gradient be maintained, right? So what will happen once these will flow through? Let's look at the two main types of channels, the voltage gated channel and ligand gated channel, right? So what will happen is that once there is a particular voltage that is required for these ions to move through, right? For example, we say that a positive like if the voltage is between minus 50 millivolt to plus 30 millivolt, this channel will be open. Right now, say that a lot of positive ions have moved across it, right? Such that the voltage has become plus 32 millivolt, right? So automatically the channel will close, right? It will no longer be remain open. Similar for ligand gated channels, as soon as this chemical molecule or Q is gone, right? It is finished. These channels will close. It will no longer remain open. So that is how they will first stop the transfer or the movement of ions, right? Secondly, what they will do is, so this effort will still won't maintain the gradient, right? It will simply halt the flow of ions any further. Now, to maintain the gradient, what do they have to do? It will be maintained by ion pumps that will actively push the ions opposed to their concentration gradient to maintain a particular charge inside and outside the membrane of the neuron, right? So what will happen is uh, there will be ion pumps that will come into picture. This is the same figure that I discussed earlier in the PYQ session also. So I'm not going to go into detail of this. What it will simply do is that this pump will move the ions against their concentration gradient so that even though sodium is present more abundantly outside, it will throw more sodium outside. Even though there are more potassium inside the membrane, it will throw more potassium inside, such that this difference is maintained, right? And since it is doing it against their actual concentration gradient, it will require ATP or energy, right? So it will consume ATP to actually perform this function. So this pump consumes energy in the form of ATP to maintain these gradients, and they account for two-thirds of all the energy that is actually used by neurons. So they are very, very energy consuming pumps, right? So we will begin with this basic electrical parameters in the neurons, wherein we will compare basically the neuron to an electrical circuit, to a very basic electrical circuit, and we will keep adding different components into this electrical circuit and see how that is correlating with our neuron and how signal is transducing into our neurons also, right? Just give me a moment. Yeah. Yeah, I'll just share my screen again. So as I said, what we are going to do is we are going to understand the basic, a very basic kind of electrical circuit, and we are going to correspond or correlate how basically signal is transduced within a neuron uh, by understanding how such an electrical circuit is actually working, right? So when we talk about current, what is current? It is basically the flow of charged particles, right? What are these charged particles? They are electrons. And when we talk about the flow of these charged particles per unit time, that corresponds to current. It is denoted by the symbol I, and the unit of this current is amperes, right? In neuroscience, we usually deal with amperes in the current in the order of nanoamperes and picoamperes, that is 10 raised to power minus 9 ampere or 10 raised to power minus 12 ampere, right? And the direction of the current, what will it be? It will be the same as direction of flow of positive charge, and it will be opposite to the direction of flow of negative charge. Now, these are the very basics that we have probably, understood for the first time when we were in maybe seventh, eighth, or 10th, right? So in physics at that time. Yeah. Next, let us also recollect what voltage is, right? So the electric potential difference is known as a voltage. That is the difference between 2 points. Difference between the charge in 2 points is actually going to be known as a potential difference or voltage. So it is always measured between 2 points. That is, it is a relative quantity, and it will be the driving force for the current, right? So there will be a difference in these in the charge between these 2 points, which will be what is driving the current from this positive charge plate to this negative charge plate, right? Also, it will be denoted by V, and the unit of voltage is in volt. In neuroscience, we will get it in order of millivolts or 10 raised to power minus 3 volts, right? So if we talk about the membrane, the inner side is the cytosolic side and the outer side is the side outside of the neuron, the exterior, right? There will always be a membrane potential between these two sides also, right? So why? Because the inside over here is negative and the outside over here is positive, right? So this will be the cytosolic side, this will be the extracellular side. Then we talk about resistance. What is resistance? It is basically the impediment to flow through the membrane, right? It is denoted by R. The unit of resistance is ohm. And there are various channels in the membrane which can conduct current and have different resistance values. In neuroscience, what is the order of resistance that we deal with? Because we are dealing with very, very tiny objects, the resistance is amplified over here, right? So the resistance that we deal with is mega ohm or giga ohm, that is 10 raised to power 6 ohms or 10 raised to power 9 ohms, right? And what we are saying is between this inside and outside, there are different kinds of potentials that are present. Inside is negative, outside is positive, and between them that there is a resistance, right? The resistance could also be the ion channels that are closed, the narrowness of these ion channels, etc. Right? The membranes not letting the ions, the charged ions pass. All this is resistance. Okay. So uh, let us draw an analogy with the water flow, right? So if we talk about the hydraulic pressure difference, that is the going to be the voltage difference. That is the difference in height between this first object and the second drum, right? This first and second drum, the more the height difference between them, the higher the voltage difference, the higher the hydraulic pressure difference, right? The charge of the flow will be the quantity of the water that is flowing. Current will be the rate of flow of water, and the wire that is connecting these two drums could either be a conducting wire if it is very thick and it has a lot of area, or if it is very thin and it has very less area that it can flow through, that water can flow through it, it will act as a resistor, right? Then flow is proportional to the pressure difference. That is, V is directly proportional to I, right? And flow is inversely proportional to resistance. So I is inversely proportional to resistance, or it is proportional to 1 by R, which gives us this value V is equals to IR, or I is equals to V by R, which we know is the formula for current and voltage respectively, right? So the SI unit over here generally is 1 volt is equals to 1 ampere into 1 ohms, right? But in neuroscience, like we said, there are different orders that these things are present in. So it will basically um, we can extrapolate that those values that we have discussed will be more commonly present in case of neuroscience. When we are studying the subject, it will basically account to 1 millivolt is equals to 1 nanoampere into 1 mega ohm, right? Then let's talk about conductance. So we talked about resistance before this, right? Resistance is an impediment of current to the flow of current. Conductance is the reverse of that. It is how smoothly the current is flowing, right? The ease with which the current can flow. It is denoted by G. G is equals to 1 by R, or it is denoted by ohm inverse, or another unit that is known as Siemens, right? Siemens. Then I is equals to V upon R, or I is equals to VG, right? Also, the more the conductance, the more the current will flow, right, for a given voltage. What is the advantage of having conductance rather than resistance is that when conductances are kept in parallel, they add up, right? Then the capacitance comes into picture. What is capacitance? Capacitance is basically the capacity of a particular membrane to store charge, right? Two conductive layers are separated by a dielectric or insulator. So there is a dielectric that is present over here, and the charge will accumulate on both the ends of this insulator or dielectric, right? It is denoted by C. The formula is C is equals to Q by V. That is, it is equals to charge upon voltage. The unit of this particular capacitance is farads, right? And in neuroscience, the relevant unit is picofarad. So the capacitance that we actually obtain when we are talking in terms of neuroscience is in the order of picofarads or 10 raised to power minus 12 farads. Right? The symbol of capacitor is only a little different than the symbol of this potential difference of voltage. Right? In case of voltage, since there will be a difference between voltage at these 2 points, it will always be one bigger line and one smaller line. While in case of capacitor, equal charge will actually accumulate at both the ends. So the symbol will be two equal lines, right? Then if we include this in the analogy of water flow, what will we get? That the water pressure difference was the voltage. The water flow was depicting the current. The amount of water was depicting the charge. And the narrow pipe was depicting the resistor. Now, what will be the capacitor? If we add a rubber membrane between this water pipe, what will happen is that water will flow from one end, right? The particles of water will not be able to pass to the other end, but like still a flow, like the flow will be maintained, right? So current can flow through the capacitor even when no electrons move, right? So when there is no charge or no water flow also, current can actually pass from this end to this end, right? Why? Because it is expanding a little. It is that rubber is not remaining like this. It is expanding a little. So it is allowing current to flow. But current changes or charge stored on the capacitor, right? When the capacitor is charged, it creates an opposing voltage. After a while, the opposing voltage will be equal to applied voltage and current flow will stop. So it will not continuously, be deforming and going, this rubber will be going into this shape. After a part, charge will accumulate on this end, right? And it will create enough resistance for it to flow like this, right? So the capacitor, there will be no more current flow in this capacitor after a while. And this capacitance tells us the value of how much charge can be stored, right? For example, if that will be dependent on this dielectric, right? For instance, it could be possible that 6 volt of charge can be stored, or 9 volt of charge can be stored, right? Okay, so when we talk about capacitance in a neuronal membrane, it is in basically parallel to the resistance, right? So what will be the capacitance over here? If yeah, so resistance is inversely proportional to the surface area, right? Because if there will be a smaller surface area, there will be more resistance, more surface area, less resistance, like we also again did talked about in the previous year questions, right? The first or the second question, I guess. Then capacitance is going to be proportional to the surface area. The more the surface area. So for example, I have this big of a capacitor, it can store more charge. If I have only this big of a capacitor, it can store only charge corresponding to this much area, right? So that is going to depend or vary, right? So sometimes we talk about capacitance and resistance per unit area. What we are going to see is uh, we are going to talk about how these are related, and most of these studies that we are going to talk about have been done in this particular organism. squid, right? So for these kinds of studies, squid is a good model organism, has been a good model organism. And most of these studies were done by Candle, who is again a Nobel laureate for these studies that he has performed on the squid giant axon. So what is a squid? It is a marine animal. It is one of the model organisms in neuroscience. Hello. I think uh, the video got interrupted. I hope I'm audible now. So my connection looks stable now. I think there was a trouble with the connection because of the weather over here. Yeah, so I'm going to share my screen again. I'm going to go through this particular slide again because I don't know whether I could get through with it or not. Get this through to you guys. Yeah, okay. So what I was saying was the squid is one of the model organisms in which a lot of these studies have been done regarding the action potentials, how signal transduction is happening across neurons. So it has a squid giant axon, which is the large axon of 1 millimeter of diameter, which is relatively easier to study than microns, small axons. So that is why it is one of the model organisms, right? And Candle was one scientist who has done a lot of research. His research about these phenomena that we are going to discuss about in the remaining session on the giant squid axon, and he is actually a Nobel laureate who has received a Nobel Prize for these studies, right? So let us talk about the iron concentrations in the squid giant taxon, right? So like humans, the intracellular concentration of potassium in the squid is also very high as compared to the extracellular concentration. Similarly, the extracellular concentration of both sodium and chloride are much higher in them as compared to the intracellular concentration, right? The resting membrane potential of a squid axon is around minus 65 volt. In humans, it is at around minus 70 volt. So that is also quite close, right? Also, studying this particular animal will also help you understand like how it might have been decided that okay, these are the two ions that are going to have more concentration outside, this is the ion that is going to have more concentration inside, right? Because we have all actually originated from this marine life, right? That biological soup that was present after the Big Bang, from which all of us have actually originated. And at this time, when there were marine organisms that are all our ancestors, right? They have always lived in an environment where NaCl is more in the outside environment, right? So they had to have some other cations that are present in more number within the cells. So that was chosen to be key positive. It might have been more abundant inside the cells at that time, or it was maybe selectively chosen over magnesium because it was a monovalent ion. Something of that sort might have happened, right? So this is a theory. So yeah. Uh, at rest, what does the membrane potential look like in neurons, right? So again, this is the outside wherein there is sodium, calcium, and chloride that are more concentrated, and this is the inside of the cell, wherein potassium along with other anions or negatively charged ions are actually more concentrated, right? So this is how it looks like. When we talk about this membrane potential within the neurons, we can basically quantify it using electrodes, like the ones that are shown over here. So for example, if we talk about this electrode, both of them over here are outside, right? At this point, what will it look like? Since both these electrodes are outside in the extracellular environment, the value will be zero, right? As soon as it is dipped into a nerve cell, it will start this oscilloscope, which will give a reading like this, like this. This can be like an oscilloscope display. So as soon as it is dipped inside a nerve cell or a neuron, this is what it will show. Like inside that nerve cell, with respect to outside, is a negative potential, right? And that is what is depicted over here. So oscilloscope will look like this, and the reading will actually be like this only, like it is shown over here. So it could be like this, then maybe this. So this is an oscilloscope reading, right? Yeah. Okay. Also, not just to read the potentials that are actually present in the cells, such apparatus and electrodes can actually be used to stimulate the neurons also. So if we stimulate the neurons such that we are giving negative current into the neuron, right? So if we are, it is already negative inside, right? If we are giving more negative current, it will go further negative, further more negative, right? Like we were talking about. And this further more negative value will be known as hyperpolarization, because it is at a negative value, so it is polarized. If it is going towards a positive value, like we will see in the next slide, it will be called as depolarization, because it is going reverse of what was actually there. If it is going at a more negative value, it is going to be hyperpolarization, more polarization, right? So over here, there are two electrodes. One is actually measuring the voltage, because it has this oscilloscope attached to it, and this is the recording from that measuring electrode. And the other electrode is actually passing negative current because it is connected to a current generator, right? So this is what the graph will look like. If we were passing positive current instead of negative current, like as I said, there will be depolarization or reversal of this polarization. That is, the value will start going more towards the positive side, right? And this is what it will look like. So we will call it as the membrane being depolarized. Yeah. So the question is that what will determine the speed of change of this membrane potential? Why do we ask that question in the first place? Is because even if we are giving a current, the value is not changing that much, right? So look, let us go back and look at how much current we are giving and how much the value is changing. Ignore this action potential value for something, right? Because as I said, we will discuss action potential and why that particular peak looks like that in the subsequent lecture, right? So over here also we are giving so much current, but the value is dipping only a little bit as compared to the amount of current that we are giving. So why is that happening? Right? What is determining the speed of change of the membrane potential? Right. So we are going to talk about these two major phenomena with corresponding to this. They are the time and the length constant. So let us begin. First, let us look at the charging or discharging of the membrane. Okay. So let us first look at this circuit again. It has the inner side, the outer side, the resistor and the capacitor. Right? What we are saying is that we are charging the membrane. That is, the current is flowing from inside to outside over here. Right? So initially what will happen is that once it starts flowing, obviously resistor is going to resist the current from flowing. Right? So all current will flow through the capacitor. What will happen? Charge will start accumulating on this capacitor, right? So initially more current will flow through capacitor and less through the resistor. As the capacitor develops a voltage, current through resistor increases. So as it develops or collects or accumulates charge over here, then voltage will also start forming, right? So after voltage starts forming over here, this has some voltage and this has zero voltage. So now current will start flowing from the resistor. It will change its path, right? So some current will flow from here, some will flow from here. Then current through capacitor will decrease. More and more current will start flowing from the resistor and less through the capacitor, right? And the current through resistor will increase. Eventually, all current applied at I will flow through the resistor and the capacitor will reach a stable voltage, which is known as IR, right? So eventually, all the current will flow through this resistor and none through the capacitor. And the voltage over here will become IR, right? Because there will be a difference between voltage on this end and this end, right? So then what we will do is we will suppose that the current source is now removed, right? And now there is a voltage difference that is existing at this capacitor, right? So it instead of acting like a true capacitor, it has a voltage difference across it. The discharge current will now flow through the resistor first, and voltage across capacitor will gradually reduce, right? So, sorry. So, as the current flows through here while discharging, this voltage will reduce, right? As the voltage reduces, now current can flow through here also, right? Eventually, the capacitor will return to neutral position, that is uh, no voltage difference across the capacitor, voltage will become zero and there will be no current that will be flowing, right? Because there is nothing that is going to drive the flow of that current. So what is going to be the rate of discharging of this membrane? Now that the charge was removed, this current I will flow from all of this current will flow through the resistor. Let's say the capacitor was charged to some voltage, which is now denoted as V0. Now the capacitor is allowed to discharge, right? Charge across capacitor is Q is equals to CV, that is capacitance into voltage. Let's say that the charge changes by a small amount, that is dq. Obviously, because the current is also now not applied, charge will change slowly, right? In a little time, it will change slowly. The value will be dq is equals to c dv, right? So why? Because small voltage is being affected by small charge change, right? And the rate of change of charge will be proportional to rate of change of voltage. So dq by dt is equals to c dv by dt, right? We can write it like this also. It is still same as q is equals to cv, just that small charge is changing small voltage and the rate of this charge change will affect the rate of voltage change, right? That is the only two variables that we are adding over here. So what it will look like is dq by dt is equals to c dv by dt. We also know that dq by dt is equals to minus I, right? And that minus I could be equal to minus v by r, right? Because I is equals to v by r. So minus I will be minus v by r. So thus we can say that this c dv by dt is equals to minus v by r, right? And that will basically give us this uh, equation, that is dv by v is equals to minus dt by rc, right? This rc is resistance into capacitance, and this resistance into capacitance can be written as tau, right? And it will give us this particular formula or equation, which will be v, that is voltage, is equals to v naught, or the voltage it had reached, into E, the equilibrium, E is actually a constant over here, minus T, which is a time that it is taking upon RC, which is basically the resistance into capacitance. And this resistance into capacitance can actually be written as tau, right? So this tau will signify the time constant. We will come to that. this will be an exponential decay at time t is equals to 0. V will be equal to V naught, right? As t will become larger, the fraction e is equals to minus t by tau will become smaller than 1. So V will reduce, right? So it will keep on reducing. This is while it is discharging, right? At time t is equals to infinity, V will become 0. So the entire membrane will have been discharged. Now, tau, which is the time constant of the membrane, determines how fast or slow the capacitor will charge, right? It will not depend on the area of the membrane. Why will it not depend on the area of the membrane? Because if large area is present, right, capacitance will be larger in proportion to the area and resistance will be smaller in proportion to the area, right? So, like we said, resistance in this much will be smaller, but capacitance in this much area will be smaller, but capacitance in this much area will be bigger, right? So it will be larger in proportion to larger area. Similarly, resistance will be smaller in proportion to area. So if there is a smaller area, there will be more resistance. If there is a more area, there will be less resistance, right? So that is why time constant will not depend on the membrane. Time constant will instead depend on the properties of the membrane, such as the density of ion channels, thickness of the membrane, etc. Right? Now let us talk about this time constant, that is tau. Right? So T by tau is going to be a dimensionless quantity, while tau itself will have the unit of time. And like we discussed in the PYQ session, the order of time that will be represented by tau will be in the order of milliseconds, right? So this we have discussed a while back, so I'm not going to go into detail again. At time, at t is equals to 0, again, as we said, e raised to power minus t by tau will become 0. So v is equals to v naught. And at t is equals to tau, v is equals to v naught e minus 1. So at time t, right, the value of V will be approximately 0.37. So V will become approximately 0.3% of its original value, or tau t will be the time at which V will reduce by 63%. That is, it will only remain to 37% of what it originally was, right? That is the value of tau as time, right? So when tau will be equal to t, V will have reduced by 63%, right? If tau is larger, it will take more time for membrane to discharge. But if tau is smaller, it will take less time for the membrane to discharge. And it indicates the amount of time it takes for a membrane to charge or discharge. So right now, what we were looking at is charging of the membrane, discharging of the membrane. So this is what the graph for that looked like, right? Charging graph will be exactly opposite of this, right? Over there, what will happen is that tau will be uh, uh, the amount in which V becomes 63% of what it originally was, right? So at 0, from this point to this, at 63% of what it originally was, T will be equal to tau, right? Then let's talk about the length constant of a branch of neuron. So this is what Sir was explaining in his lecture, that when we talk about conduction of electricity or any gradient across a neuron, we usually consider that there is a spherical neuron, we are injecting it at one region and it is radially transmitting across this entire surface. But usually the conduction is happening across a dendrites or across axons, which are longitudinal, right? So they look more like this. And once they are more longitudinal, at the point of injection over here, there will be the maximum current. And this current will keep on reducing as we are going farther and farther away from this point of injection, right? And this is going to depend on like how fast it is reducing is going to depend on two components. One is the membrane component and the other is the axial component. And we will shortly discuss about these two, right? The second thing that was depicted by this length constant is that uh, it depends on x, that is distance, and the length constant that over here is given by lambda, right? So at basically lambda is equals to x, the neuron, the current will have dissipated 37% of the actual current that it had been given, right? So let us talk about this length constant of the branch of the neuron. If this is the equation that is given, delta V is equals to delta V naught E minus x raised to power minus x by lambda. It is also going to be an exponential decay like we saw in case of time constant. The maximum value over here that is reached will be when x is equals to 0. So at the point where the injection is going to happen is x is equals to 0, right? At that point, the maximum value of V will be present, right? And this is denoted by the point, this is at the point of injection that is denoted by delta V naught, right? The rate of decay is determined by this parameter lambda, which is the length constant. If lambda is equals to 1, the value becomes delta V is equals to delta V naught E raised to power minus 1. Or it becomes 37% of the maximum value, right? So when lambda will reach 1, will reach unit of 1, only 37% of the voltage will remain as compared to the point of injection, right? Lambda is the length constant and it will tell how much length does V reduce by, it tells in how much length the V reduces by 63%, right? And there are two factors again, let me tell you that length constant is going to depend on. One is the RM or the membrane resistance, right? This is going to be the RM. And the other is RA or axial resistance, and this is a RA, right? So let us see what is the implication of RM. This RM is membrane resistance per unit length. If the membrane resistance is more, there will be less leakage and bigger lambda. Why do we say that? Because, for example, the resistance is more on the surface of the membrane, like then the current that is flowing within the membrane will not be able to move out of that membrane, or lesser current will be able to move out of that membrane because there is higher resistance on the membrane, right? So, since more current is staying within, the lambda or length constant will increase. The current will be able to flow for a longer distance, right? So Rm is inversely proportional to length constant, right? While Ra over here, uh, sorry, Rm is directly proportional to length constant, right? More the Rm, more the length constant. While in case of axial resistance per unit length, which is ohm by m, if there is more Ra, there will be more hindrance along the length and the lambda will be smaller, right? So what does this mean? That if the diameter of the tube that it is flowing through is smaller, right, then more resistance will be encountered and less of it will be able to move out. More of it will try to move outside the membrane, even though the membrane resistance is there, right? So that is what is going to happen. So it is going to be inversely proportional to this value that is Ra, right? So the unit of lambda over here is meter and lambda is equals to under root Rm by Ra, right? So Rm is going to be ohm by meter, ohm into meter upon Ra, that is ohm upon meter. So under root meter square, right? And once the under root is taken off, the unit is going to be meter, right? So let us look at the relationship between the length constant and the axon diameter. This is probably going to be the final slide, if not the second last, right? So Rm will depend inversely on the surface area, right? If there will be more surface area, then there will be less resistance, right? So it will vary as 1 by 2 pi r, right? RA will depend inversely on cross-section area. That is, if there will be more diameter or more radius, there will be lesser RA, right? So, RA is varies as 1 upon pi r square. So, Rm by Ra is proportional to radius. In other words, length constant will increase with radius. So as the radius of this tube increases, the length constant will also increase because axial resistance will go down, right? Also, this is significant because thicker axons will allow conduction of electrical signals with less attenuation. This is what happens in giant squid axons, right? But in our case, the radius is much smaller than the giant squid axon. So, on the other hand, what we do is that we increase the RM. Increase of decrease in instead of decreasing the RA, we increase the RM by myelinating our axons. That is how our neurons will conduct electrical signals with less attenuation, right? So time constant will be how slowly the signal is changing in a neuron, right? And length constant is how far the signal is spreading in a neuronal branch. So that is it for this part also. And thank you for sticking along. Yeah. So because there were no questions, I will just assume that you guys have no queries, right? Although, yeah, there were some queries that were present or posted in the discussion forum. I opened it this morning only, so I did try to solve some of them. One of them that I did not respond to was regarding an assessment question. Question number 8, I did not even went back and look go back and look at what that question number 8 was, because I know that that assessment is still open and I am really not allowed to, discuss those particular answers with you guys until the deadline passes. So, yeah, that is it. Thank you for bearing with me this long. I'll just stop the recording and we can end the session.